So it's my pleasure to introduce Knetminer today. Knetminer stands for Knowledge Network Miner with a silent K, and it's an AI-assisted gene discovery platform. It's part of the Elixir UK node and has been developed at Rasmussen Research and the recently established Knetminer spin-out company. So the presentation objectives, there are threefold. Want to start with giving you an idea of how we have integrated the pangenomics data from Plaza that Michiel presented just now into the Knetminer knowledge graph. Then I'll go into, I will be brave and I will go into a live demo and show you how the integrated data is used in a gene discovery platform with a specific use case around GWAS data analysis. And finish off with saying that the data that we have integrated can not only be used within the Knetminer platform, but we also provide APIs and gene pages for people who are maybe developing their own databases and would like to cross-reference into the Knetminer knowledge graphs. So with that, let's just spend one slide introducing the Knetminer knowledge graph and the vision behind it. So our vision is to try and connect as many life science and plant science data sets into a semantic knowledge graph using semantic data integration and text mining technologies. And why do we want to do that? Because we feel we think if data is integrated coherently, it will allow us to build, or everyone to build, a lot more intelligent and advanced search capabilities on top of the data. And the other reason is by integrating the data, cleaning it up, we can provide fair data that is interoperable and reusable more easily. So Knetminer knowledge graphs, they are generic. We don't focus on just barley or just one species. We are cross species. And the use cases I'll be showing today, they're around plant and crops. The query languages that we using to query these knowledge graphs, maybe you're familiar with them, they're Cypher or Sparkle based. Okay, so let's switch over now to what specifically we have done as part of the EPAM project. So once the Plaza French team have completed their parts, it was about understanding how to exchange the data from Plaza into a platform like Knetminer. So we spent a couple of sessions with them to understand the data and agree on a standard format. And what you see on the screen in this table is a format that we came up with. We said, okay, the data we want to bring into Knetminer, into knowledge graphs, is the gene presence absence variation information and the synteny scores that Michiel presented just now. And that could be as simple as just a simple table. So we've got gene IDs, we've got the synteny scores. For every gene, we record in which cultivar it is present or absent. So this gene is present in AN1 and is absent in the cultivar C24. And the orthologous, representative orthologous gene ID would be part of another column. So this data was currently prepared by the Plaza team for Arabidopsis, rice, tomato, and barley. And we decided to model it as simple as a simple triple in the knowledge graph being gene, and then the status. So is it a present or is it an absent relationship in a specific genome or cultivar? So Knetminer has capabilities and features to integrate a tabular OWL XML data into a knowledge graph using a specific ETL pipeline. So we can bring together hundreds of different data sets, transform them, enrich them into a semantic graph. Our graph database are based on Neo4j. And then once that's developed, the Knetminer app can search and visualize the data. And this will be, I will just focus really for today only on the end user bit, which is about how to search and visualize the data. If you're interested to know more about how we integrate data and the ETL pipeline, contact me offline. So for the use case and the demo, I would like to focus on a specific case story about analyzing and interpreting GWAS data. So GWAS standing for Genome-Wide Association Studies. They identify regions in the genome that can be associated or linked to specific traits, like, for example, salt tolerance. So there is a statistical analysis that is done, and this association links SNPs, okay, with phenotypes like salt-sensitive root growth, like shown in this picture. And what users then do is they filter the list based on the p-values, and they try to focus on specific peaks. They filter maybe by a log p-value of 8, and try then to scan for candidate genes that are within those regions, and then ask the question, which of those tens or hundreds of candidate genes has any link to the specific trait that I'm studying? And in this case, salt-sensitive root growth. Okay, so we've got a file with tens or hundred thousands of SNPs and their p-values, and then we want to feed that into a system that allows us to search the literature, phenotypic information, pangenomic information, just more easily and quickly, or without it taking days or weeks. So with that, I will switch over to another tab. And Sebastian, I hope you can just confirm if this is still
It's
visible.
still showing now it has switched, and I can see now Knetminer open up on the My Resources page.
Perfect, thank you. Okay, so this is our entry point into app.netminer.com. We're going to pick a specific resource. There are two that are completely for free. You don't even need to be registered to use them. If you do create an account on netminer.com, you get much higher limits to use it. And there are certain packages with a premium that are subscription based. So I'm going to focus on this knowledge graph called Plants Light. And as I said, it's all public, and you can just redo exactly the same steps after my talk as well. So for this demo, we're going to focus on Arabidopsis thaliana and go into a specific type of search over the knowledge graph called GWAS peak search. There are other types of searches. Okay, so now what we need to do is just spend a couple of seconds to populate this form. So we have conducted the GWAS study. Let's say we've used a tool like GapHit, and we have mapped our SNPs to associate our SNPs with specific phenotype, and we're going to upload this file now into the platform. So this is as simple as just being a file of SNP locations and their p-values. Has this 4 columns. I'm going to import this into press import. And now it's asking me to set 2 parameters. Which SNPs do I want to focus on? I only want to focus on SNPs that have a log p-value of, let's say, 8 or larger. And what is the SNP window, like LD window, I want to focus on? So in this region, I'm going to search for candidate genes. And just for the demo to be a bit quicker, I'm making it a bit smaller, this window. So 2.5 KB left and right of a SNP. I'm searching for candidate genes. I'm giving my experiment or study a name. So my GWAS, let's call it salt response study, just salt response. And I want to ask the question, with the genes that are significant, statistically significant, which of them has any evidence to be related to, for example, salt or root-related phenotypes, traits, gene ontology terms, and so on. Okay, pressing search now. So it scans through those list of SNPs that were in my file, and you could see within seconds it can give you an idea of which of those SNPs that were significant are linked to genes that can be related to salt or root-related traits. The table is sorted by Knetminer score, but I'm going to resort it now based on the actual SNP p-values coming from the GWAS study. Okay, so these are the SNPs that were significant, they're linked to these genes, and now I can look at these different columns and see what they mean. So the evidence that has linked it now to those specific traits can be based on text mining, it can be based on homology, it can be based on protein-protein interaction data. And then if you click on this evidence types here, it summarizes the evidence and shows you what are the different phenotypes that are linked to this specific gene. So this is a view that a lot of you are used to. It's a very classical tabular way of looking at data. But the real power of Netminer is if we start generating knowledge graphs from this tabular views. So I'm going to select now just the top candidate gene, CGL, and say create a knowledge graph. Okay, so this is an interactive knowledge graph. I can move things around, so it's all interactive. On the left-hand side, I've got a legend that explains what the different nodes and edges mean. I can select stuff. Okay, I can go select things. I can easily delete nodes, and I can add more information to the graph. I can say, now show me the cultivar information, the presence absence variation data, bring that to knowledge graph. In info box, I can click on nodes and see what they are. So we've got publications, sold, and so on. So now I can spend a bit of time moving things around, making this network look nice, which I have done already and saved. So I don't need to waste time doing it now again. I'm going to my saved networks. I'm just going to open a network that I have the same network. I've just laid out the nodes and edges a bit more nicely, so it's easier to present. And I'm going to zoom in now and just really try to focus on two things. One is the Plaza data that was added to Netminer. How is it visualized and modeled? So to do that, I want you all to focus on the specific gene that I have selected called CGL. And you can see, for example, CGL is absent in the cultivar, Arabidopsis cultivar, or Arabidopsis genome called LER. It's absent in C24, CVI, SHA. It's present in 3 of them. And you can see the node size. The node size is a bit small because it's showing the synteny score of that specific gene. So the synteny score of CGL is 0.4. And that's why that node is a bit smaller. Now, interestingly, this gene has a relationship called interacts. Okay, so I can click on a relationship. You can see it interacts based on affinity capture Western and this publication with another gene called MNS1. And MNS1 now is present in all these different genomes and has a much higher synteny score. Okay, so this is how the synteny scores and path data has been added to the knowledge graphs. Now, the beauty of it is I can analyze the pangenomic data together with all the other types of information that we can add to genes. For example, phenotypic information. I can now click on the different nodes like salt tolerance and see there's a relationship that says CGL actually has some known relationships to salt tolerance. And I can read where is that information coming from. It's coming from a specific paper that talks about CGL being more salt sensitive than wild type. And I can look at these networks and try to get a feeling for what the nodes and edges all mean and come up with a hypothesis. So we've got information about Arabidopsis on the top. Then we've got homology information that links now this CGL to orthologous genes in wheat, for example. And we can see in wheat, it has been linked to different traits like plant height and wheat leaf rust resistance, disease response. So now just to finish up, one of the latest features we've been building in the platform is called Graph Chat, which makes it all a bit easier to interact with this graph through natural language. So I can go to the Graph Chat window here and show you that you can now start typing. And Graph Chat welcomes you and asks you what type of analysis do you want to do with this specific graph. And you can now go and ask any type of question like which of the selected genes is the better target for genome editing to improve salt tolerance. I can go and select now the two genes that I want to analyze. There's two. Go back to graph chart, press enter. So it has knowledge of the visible graph, what is selected, and now it's analyzing the phenotypic data, the pangenomic information, and is suggesting that CGL1 would be the better, stronger target for genome editing, and gives you the reasons for that and the papers. You can click on the different nodes and it will highlight them in the graph, so you can fully transparently always connect the AI to the full evidence in the graph database. Okay, so with that, I'll switch back to the presentation and just say one more thing that so if you're a developer, if you're building your own databases or apps and you want to cross-reference into Netminer, we can provide the stable URLs for every gene in, for example, Arabidopsis. So the URL looks like this, and if you click on one of these links, it brings you directly to a knowledge graph with graph chat on the right-hand side that would allow your users to look at the phenotypic literature, pangenomic data that we have for all those genes in Netminer, and you can cross-reference. So Ensemble Plants, for example, is cross-referencing to this, and we're working with the Plaza team to cross-reference back to Netminer and from Netminer back to Plaza going forward. Okay, so that's I'm mostly done with my talk. So takeaway messages. Hope I can show that Netminer stands at the intersection between pangenomics and functional biology, and it is a really useful tool to help users understand complex traits across diverse species. It's an integrated data ecosystem. It provides programmatic discovery via APIs, and it can help you to build accelerated breeding pipelines. The team behind Netminer is on this slide, and Arne is part of the session as well, and also happy to answer any questions. Thank you very much. That's it from me.